Transcription. This is a process in which cells use double-stranded DNA to make single-stranded mRNA, which will later be used to synthesize proteins.
Transcription can be separated into three main steps: initiation, elongation, and termination.
During initiation, the enzyme RNA polymerase attaches to a transcription factor located on the DNA. Transcription factors attach to promoters specific sequences of DNA, which include the Tata box.
The RNA polymerase then separates the strands of DNA, creating a bubble of space to keep the strands apart.
The polymerase also lays down nucleotides that complement the guide strand of DNA, with the same combinations except for uracil instead of thymine. The new strand of mRNA will run from 5' to 3'.
The next step, elongation, is when the polymerase continues to go down the strand of DNA and lays down nucleotides. Sometimes more than one RNA polymerase transcribes at a time.
The last step, termination, occurs when the RNA polymerase reaches the terminator, a specific sequence of DNA, and drops off.
The resulting pre-mRNA, however, is still not ready to be used and will go through RNA processing. A methyl cap composed of guanine attaches at the 5' end, and a polyadenine tail is added to the 3' end, both of which are used for protection from harmful hydrolytic enzymes found outside the nucleus.
In a process called RNA splicing, certain parts called introns and extrons are removed and reattached.
These exons can be rearranged or excluded as necessary, creating different nucleotide orders. This allows for a variation in protein synthesizing codes.
Introns are sequences of DNA that are not unstable, and they will be cut out. Exons are the sequences that will be put together and used in translation.
Now the mature mRNA is ready to leave the nucleus and continue into translation.
Translation is the synthesis of proteins using the genetic code in mRNA. The main steps have the same names as transcription: initiation, elongation, and termination.
The initiation of translation step begins with a small ribosomal subunit attaching to the mRNA. The initiator tRNA attaches at the start codon AUG with the anticodon UAC to begin translation.
The large ribosomal subunit then sits over the mRNA and initiator tRNA. There are 3 sites in the large subunit: the E, P, where the initiator tRNA is, and A sites. Both the large and small ribosomal subunits are made of proteins and rRNA.
Next is the elongation step of translation. tRNA enters the A site with the complementary anticodon. tRNA has an anticodon that complements only one codon on the mRNA, along with the amino acid attached by the enzyme aminoacyl synthase.
Peptide bonds are formed between the amino acid group on the A site tRNA and the carboxyl group on the initiator tRNA. The carboxyl group is removed and attached to the amino acid group, adding to the polypeptide chain.
Through translocation, the positions of tRNA are moved left and the initiator tRNA is placed in the E or exit site and floats away. This process continues until a stop codon attaches and reaches the termination of translation.
In termination, a stop codon enters the A site and hydrolyzes the bond between the tRNA, P site, and polypeptide chain. Then, the ribosomal subunits detach from the mRNA.
Although the polypeptide chain will naturally fold as it is developed, it is still not ready to be used as a protein.
In post-translational modifications, sugars, lipids, or phosphate groups may be attached. Enzymes called chaperones may remove amino acids from the leading side of the chain.
These modifications are important to activate the protein. At this time, the more detailed tertiary and quaternary structures of the protein are assembled.
Transcription and translation are just 2 of the many processes that keep us alive and allow us to grow. Understanding these processes can help scientists make new discoveries and understand ourselves.